Hello, everyone, and thank you for joining us today as we're discussing research with the scientists within the Mind Project to cure paralysis. My name is Dalton Dietrich, and today I'm happy to introduce Dr. Pablo de Rivero Vaccari, who's an assistant professor in the Department of Neurological Surgery, an extremely important member of the Mind Project to cure paralysis. Pablo, thanks for joining us today.
Thank you. My pleasure.
So, Pablo, your research interest and clinical interest are widespread, but they really do focus on inflammation. And we've talked about inflammation many times in terms of being a very important secondary injury mechanism after brain and spinal cord injury, and the development of new therapeutic interventions to target that inflammation. So can you tell us a little bit about your research?
Yes, I've been studying inflammation for about 15 years, mostly in the central nervous system after injury, and more recently after diseases like Alzheimer's and multiple sclerosis. But basically, my work focuses on the molecular mechanisms associated with inflammation. Generally, when people think about inflammation, they think about swelling. When you see something that is swollen, you say that's inflamed. And that's definitely a characteristic associated with the inflammatory response, such as swelling, redness, heat, and pain. However, when I refer to that I study about inflammation, it pertains to the molecular mechanisms associated with an immune response that leads to fighting infections or fighting diseases, and that when it is exacerbated, it actually results in cell death or tissue destruction. Mm-hmm. So within the inflammatory response, there are two phases. One is the innate immune response, which is followed by the adaptive immune response. Particularly, I studied the innate immune response, and within that inflammatory phase, I studied the multiprotein complex known as the inflammasome. The inflammasome, it's interesting because it activates a key inflammatory cytokine called interleukin-1 beta. And interleukin-1 beta for many, many years, since the 70s, late 70s, it's being associated with several diseases, not just autoimmune diseases, but also traumatic events and traumatic brain injury, stroke, as well as spinal cord injury. So my research has two different aspects. One is to identify therapeutic interventions that can target the inflammatory response. And the other one is to look at biomarkers that can be used to monitor disease severity, disease progression, as well as can be used to determine if patients are going to do well or not following a trauma or following a disease. So when you think about how we can treat inflammation to improve outcomes, we know that overall, when we treat inflammatory response in most diseases, because most diseases or conditions have an inflammatory component to it, that we know that when we treat inflammatory response, patients start feeling better. So that makes inflammation a very attractive target. The way I like to describe it in very simple terms is that imagine that you have a paper towel that is wet. That wetness in the paper towel, that's the inflammatory response. Now, on top of that, if you put a stress, that paper towel is going to break. So that breakage in the paper towel, that's the cell death that follows the inflammatory response. So basically, with my work, I try to keep that paper towel dry, if you want to say it that way.
That's a very interesting way to present it. Inflammation is so complex and affects so many different types of neurological processes and injury mechanisms. The other area that I think is important is how inflammasome or inflammation affects many types of neurological and human disorders. So yes, you've worked on spinal cord injury, but you've worked on other types of neurological conditions as well. Can you tell us about some of those?
Yes. When I first started my life as a scientist, my very first work, which is a work I got my Ph.D. for, which I did at the Miami Project to cure paralysis in association with the physiology department here at the University of Miami. We looked at the inflammasome being activated after spinal cord injury. And that was very interesting because prior to us showing that inflammation was activated after a spinal cord injury, nobody thought of this inflammatory complex being present in something that was a sterile inflammatory event. So in spinal cord injury, there's no infection. But prior to us showing that inflammation activation was present after spinal cord injury, everybody had shown actually that the inflammation was present in viral infections, bacterial infections, fungal infections. So we showed that neurons were key cells involved in the production of the inflammatory response, which is not normally what you think of when you think of neurons. You know, thinking of neurons as inflammatory cells, it was quite a novel idea. Now more people have shown that indeed neurons can secrete these cytokines that are activated by inflammasome. So that's very nice to see that this work that other people have continued with this work. Also, another project that I looked at is in the field of reproductive senescence in a collaboration with Dr. Raval in neurology. We looked at menopause or reproductive senescence, and we showed that that the female reproductive system secretes these vesicles that we refer to as extracellular vesicles or exosomes. They are secreted from the reproductive organs during menopause, and then they travel in the bloodstream. They cross the blood-brain barrier, and then they go all the way to the brain, inducing an inflammatory response. So that's a mechanism by which the reproductive female organs actually communicate with the brain to induce an inflammatory response that we already know that is present as a result of menopause. The interesting thing is that in this case is mediated by exosomes. Normally, when you think about the heightened inflammatory response in reproductive senescence, is due mostly to hormonal imbalances, which obviously that's a big part of the issue. But with this project, we actually showed this very interesting involvement of extracellular vesicles. Also, looking at biomarkers after traumatic brain injury, we were able to show that if you collect samples from brain injury patients within the first 3 or 5 days after trauma, and you measure the levels of inflammation proteins in the cerebrospinal fluid and also the serum of these patients, those patients that had higher levels of inflammation proteins acutely after trauma were also the same patients that did not do well when you look at them chronically 5 months after injury. So you can actually measure inflammation proteins in patients acutely and then predict if those patients are going to do well or not under current treatment paradigms. Also, another project I like a lot, which was in a collaboration with Dr. Nancy Brackett and Imad Ibrahim, is that we looked at inflammation in male infertility from spinal cord injury patients. And the cool thing about that project was that if you know, after a spinal cord injury, there's male infertility, and that's the result of sperm cells basically not swimming that fast or that well. So you can actually have the sperm of these patients in a tube. You inhibit inflammasome activation in the sperm of these patients, and then all of a sudden you start seeing that just with your own eyes, without much technology, you see in microscope that the sperm cells start swimming just by inhibiting inflammasome ex vivo in the tube. So then you can actually, the idea is that then you can actually use inflammasome inhibitors either given orally to patients or treated in the lab to then use these sperm cells for in vitro fertilization or things like that. And now nowadays, another topic that we're working on is the concept of aging or inflammaging and inflammasome activation. So inflammaging it's the concept that as we age naturally, our body starts developing a basal level of inflammatory response. And that basal level actually makes you prone to other diseases like Alzheimer's, Parkinson's. Obviously, as we age, we are more prone to getting diseases. So if you inhibit the inflammatory response that may prime you to the development of diseases, then you have a shot at actually preventing some diseases or delaying the onset of diseases. So we're trying to find ways to inhibit inflammation so that we can delay disease onset or improve the quality of life that gets diminished as a result of aging as well.
Oh, thank you very much for reviewing all of that. It's just amazing how many different types of topics, touch upon inflammasome signaling and inflammation in general. And that's why it's just a very exciting area of research, and congratulations on all the work you and your colleagues have been doing. Okay, so you're a basic researcher, basic science, translational scientist. Now you said, okay, this is important in so many human disorders. How can I use this information to actually develop new therapeutic or diagnostic approaches to actually translate this to the clinic? And I know you and your colleagues have been very active in that regard as well. So can you tell us about, some of the translational studies that you're doing?
Yes, I consider myself blessed enough or lucky enough that these days I'm part of an effort to translate the discovery that I got my Ph.D. for all the way to clinical trials, all the way to patients. So as part of my dissertation work, I had we discovered that that we can use monoclonal antibodies to treat to inhibit the inflammasome. So the exciting part is that these monoclonal antibodies, which are also known as biologics, as you might have seen commercials everywhere about drugs like Remicade, Humira, that are very are used to treat things like psoriasis, Crohn's disease, multiple sclerosis. So this drug that we're developing that we started first to test in spinal cord injury back in 2008 targets the inflammasome as opposed to other drugs that are in the market for other pathways. But we have shown that this drug, which is under the name of IC100, can be used to treat inflammation and improve outcomes in patients or in animal models of spinal cord injury, brain injury, stroke. More recently, we've shown it in multiple sclerosis. We're getting nice data also in even renal diseases. Mm-hmm. So it's very rewarding to have the opportunity to not just discover something in the lab, but going through the effort and be given the opportunity to actually take those inventions forward to clinical trials. Obviously the process is not easy, but it's very rewarding. It takes, in science we have this, we have the mantra of publish or perish. When we actually try to translate something towards the clinic, then we also have to patent. So because if we don't patent, your inventions will perish as well. So and it's part of patenting. You need it as a natural cause of trying to protect your invention, your intellectual property, so that other people don't infringe in it, so that investors are more confident of that they can actually have the return on investment once they have this huge inflow of money going into the developing of the drug. So fortunately, we've patented quite a few inventions around inflammasome in the field of therapeutics as well as the field of diagnostics. So we're excited that hopefully within the near future we'll see this IC100 being tested in humans, which would be one of my scientific goals that I'm yet to achieve. So that'll be very rewarding. It's very challenging to have to just develop something and then actually take it to clinical trials. Mostly it's the funding issue. When you think about people think, okay, so we do the research with grants. there's so many scientists applying for grants. And by the nature of how the granting process works, sometimes grant funders, they want to make sure that that science is feasible, so that they put money where they think they will get results. The challenge is that on the other side is that when you have to patent something, you have to patent something that it's not obvious. Because if it's obvious, then by patent law, it's not patentable. So that's kind of like a discrepancy between obviousness and innovation. So that's why you need to, that's when external funding agencies like private funding agencies are very helpful to the developing of drugs that eventually make it to clinical trials. To make the challenge even worse, there's this issue that grants will get you this far, but you need to get this far, So basically, there is for an investor to actually license one of your inventions. Most of the time, they want to have lower risk. They're willing to pay more money for lower risk, and the way to lower the risk is by developing that drug as much as possible, so that then they are more certain that they can actually take inventions forward all the way to patients and eventually to commercialization. The issue is that to go all the way there, from here, there is a space that needs to be funded. That's some people refer to it as the valley of death, because if you don't get funding for this small space that is actually very expensive, then that invention that you came up here with won't make it to patients. So and these are things like actually the phase of drug development, showing that your drug is safe. So showing that your invention it's safe not in just rodents, but also in non-human primates, and that it's putting part of an IND package or an investigational new drug package that then you can present to the FDA, which is what investors want to see to actually move inventions forward. So that's the challenge, but it's a lot of fun to be a part of these efforts, and I consider myself blessed that I get the opportunity to do it.
Fantastic story. Yes, it's so many stages in moving a discovery to the clinic, and many times we didn't go to school to learn how to do that. So it's very challenging. We're fortunate that you have and others have colleagues outside in the industry field that can actually help us and guide us to move these things forward. So congratulations on all that. So we're talking today. You're at home, I'm at home, and we're basically social distancing and trying to get our work done during the COVID-19 pandemic. So how are you doing during the pandemic in terms of your research efforts? And is anything we've talked about today related to COVID-19 therapies or diagnostics?
Yes, actually, these days I've kept myself busy doing a few things, or more than a few things, and one of those things have been actually keeping track of the science that comes out regarding COVID-19. As many people may be aware, there is this heightened, exacerbated inflammatory response as a result of in some patients that die actually with COVID-19. This inflammatory response is referred to as cytokine storm syndrome, where you have a variety of inflammatory cytokines, some of which are activated by the inflammasome, such as interleukin-1 beta, which goes on to activate interleukin-6 upon stimulation of the liver. So an IL-6 is actually a key cytokine that correlates well with death in COVID-19 patients. So it is so the inflammasome presents itself as a good target to study COVID-19 in these patients. We've shown ourselves that in the lung of rodents with after brain injury, they develop acute lung injury, and this acute lung injury is similar to the events that you see in patients with COVID-19. So there's an involvement of inflammasome in COVID-19, in the cytokine storm, and the and the effects that you see in acute lung injury or the level of the lung. So definitely, the inflammasome is an attractive target for it. So I've been keeping up and see what where we can use our expertise to help move those efforts forward, because obviously we all are in this together.
Well, Pablo, thank you very much for spending some time with us today and talking about inflammation and the inflammasome, which is such an exciting area of research, and hopefully it is going to make a difference in people's lives one day in terms of targeting this cell signaling cascade. We'll certainly have you back in the future, but congratulations to you and your colleagues for everything you're doing to move science forward. Thank you.
No, thank you. Thank you for the help. It's always great to be part of this team.